The way neurons communicate between one part of the body and another part of the body is called an action potential, or it's also called a nerve impulse. And it's how they transmit information from the dendrites down through the axon to the synaptic end bulbs. So if we take a look at this here, we've got the cell membrane of our neuron. And the cell membrane of a neuron is excitable, meaning that it's kind of like a charged battery that can be used in order to do something. So what happens is that ions, and remember ions are charged particles like sodium and potassium, can cross the cell membrane through these special channel proteins which create like a pore or a passageway for them to get in and out of the cell membrane. And these pores or these channels can be closed and opened, and the opening of the channel can be triggered by something like a neurotransmitter or a chemical binding to it, which is how we taste. It could be mechanically gated so that something physically pushes it open in order to allow those ions to move across the cell membrane. So let's take a look at some of the steps associated with a nerve impulse. So first of all neurons have a membrane potential. And if we look here, we're looking at the membrane, the cell membrane of a neuron. We've got the extracellular fluid out here and the intracellular fluid in here. And in our drawing here, this is a voltmeter or voltometer, which helps to measure the difference between the voltage out here and the voltage in here. And this voltage is determined by the distribution of ions across the cell membrane. So basically, if it's positive outside the cell and negative inside the cell, it means that there are more positively charged ions outside the cell than there are inside the cell. Now, this membrane potential only exists around the membrane. And as we mentioned, we can kind of compare it to a charged battery. So you're probably familiar with batteries. Think of like a AA battery. It's got a positive end and a negative end. And those have to be lined up properly when you put it into a toy or something in order to make it work. And so this difference here has to exist in order for a neuron to have a nerve impulse. So let's take a look at what we call the resting membrane potential. So this is the state of the neuron cell membrane when the neuron is not having a membrane potential. But it's like a charged battery, it's ready to be stimulated, but it's not currently being used. So there's something called a sodium-potassium pump, which is not shown in the picture here, but it is a protein that goes across the cell membrane, and it actively pumps or moves sodium, which is Na+, out of the cell, and potassium, which is K+, into the cell. Now, you may be confused because both of these are positively charged, whereas down here we're talking about positive and negative charges. Well, the key is that more sodium is pumped out of the cell than potassium is pumped into the cell, and these two charges are relative to each other. So you can think of this negative as also kind of meaning less positive. So relative to the outside, there's more positive sodiums outside than there are positive potassiums inside. So at rest, the resting membrane potential of the neuron has a difference in charge of about negative 70 millivolts, so more negative inside than outside. Now, in order for the cell to respond when it's excited, it goes through something called depolarization. And the primary thing that depolarizes a neuron is sodium. So as I mentioned, there are channels. These are those proteins that are embedded in the cell membrane, and they can be gated, meaning that they can be closed or opened, and a stimulus can close them or open them. So for example, some gated sodium channels open when a neurotransmitter binds to it. Or it might be the energy and heat that opens up the sodium channel. or the movement of air might open up the sodium channel, which would trigger something like your ability to hear. A chemical, like the chemicals in your food that you taste, might bind to a gated sodium channel and open it up. But regardless of what opens that sodium channel, what happens is that sodium, starts to diffuse into the cell membrane of the neuron. And so when this happens, the inside voltage of the neuron becomes more positive, and the nerve impulse has started. So let's go back a slide and take a look at this picture here. So remember the resting membrane potential, there's more sodium outside the cell than there is inside the cell. And the outside of the cell is more positive, whereas the inside of the cell is more negative. So pretend like there is a protein here, a sodium channel, and a neuron binds to it, and it opens up that sodium channel, and now this positively charged sodium starts going into the cell. So now this inside is no longer less positive because we're adding positive sodiums to the inside. And so what happens is that the relative charge actually changes, and at the end of depolarization there's actually more positively charged ions inside the cell than there are outside the cell. And that's how we get to that positive 30 membrane potential here. All right, now depolarization doesn't happen to the entire cell all at once. It only happens in one small area, and then it gets propagated or transmitted along the neuron. So remember we said earlier that information in a neuron always starts at the dendrites and it moves down through the axon towards the axon terminals. So if, let's say, a neurotransmitter binds to a dendrite here, it can cause depolarization in the dendrites. Now that depolarization can open up another a gated sodium channel near it. And so as this area depolarizes, it causes this area to depolarize. And then this area will open up sodium channels next to it and cause it to depolarize. And then that sodium channel will open up sodium channels near it, and sodium will go into the cell, and that area will depolarize. And this happens all the way along the axon, down through the axon terminals into the synaptic end bulbs. So they call it a wave of depolarization because it's depolarizing in one area and then moving down the neuron to the other area. So after depolarization, a series of events happens where the sodium channels close and potassium channels open, and there's a movement of potassium and a whole bunch of stuff happens. We're not going to go into all the details, but the end result is that the resting membrane potential gets reset. So basically the sodium channels close, sodium gets pumped back out of the cell. You've got your sodium potassium pump constantly pumping sodium out of the cell and potassium into the cell. And so after the nerve impulse happens, the resting membrane potential gets reset and the cell is ready to be stimulated again. So basically a neuron can send a nerve impulse, and then once that the membrane potential is reset or recharged like a battery, then it can have another nerve impulse. And so as long as the stimulation keeps coming, the nerve can keep responding as long as it has time to reset the resting membrane potential.